I'm really excited, and Sarah Batterman and I are really excited to introduce Dr. Mark Ritchie today from the Department of Biology at Syracuse University. And we want to say hello to everyone online who's streaming in virtually. And there is an opportunity to ask questions at the end, so please submit your questions through the Q&A box on Zoom. And Dr. Mark Ritchie received his PhD from the University of Michigan, and then his postdoc, he did his postdoc work with Dave Tillman, as some of you may know who that is, at the University of Minnesota. And then he went on to be a faculty member at Utah State before settling in at Syracuse. And his work is really, really impressive. It's ranging a huge range from looking at fractal geometry and how it affects biodiversity to thinking about global change effects on plant communities and then herbivore interactions. And now finally thinking about carbon accounting. And Sarah and I were really excited to invite Mark because he has not only a really interesting perspective on this new pool of nitrogen fixation, which we're personally very interested in, but also the way that he looks at it from a holistic perspective and then also from a top-down perspective. And so without further ado, we'd love to hear what you're working on, Mark.
Okay, so my mic is on, correct? Okay, so yeah, I'm super happy to come here. I've heard about the Carey Institute my entire career that's now longer than I wished it was. And so excited to come visit and excited to talk about some of the work that I've been doing. All the work that I'm going to talk about was not just mine, but the results of multiple collaborations of various ideas or doing field work or whatever, including Professor Jeffrey Soka from Sokoine University in Tanzania, Nancy Johnson, who some of you may know from Northern Arizona University, who's an expert on mycorrhizal fungi, but she's the one who actually suggested that I measure nitrogen fixation in grasses. I thought she was muy loco, but turned out she was right. Ramesh Reina is a plant molecular biologist who helped me do some of the molecular work. And Neha Mohan Babu was a PhD student of mine who's now doing a postdoc at Minnesota. And Emilian Mayamba is the man who can do it in Serengeti National Park. He's probably their top naturalist, but he can also take Land Rovers apart and put them back together. So the ultimate guy. All right, so what I'm going to talk about has to do with trying to increase our understanding of nutrient budgets in really dynamic ecosystems. And one of those kinds of ecosystems, as terrestrial ecosystem, is savannas, which are a mix of grass and trees. And they're always under this sort of tension in the sense that if certain kinds of factors are one way, they tend to turn into woodlands. And if other factors are more prominent, they tend to turn into grasslands. And so there's always this kind of tension between the two. And they're also, in some places, home to what we call the greatest concentration of charismatic megafauna, African wildlife in particular, in African grassland savannas, but they're also even in more depauperate faunas in North America or Asia or Australia. They're still often at that sweet spot where they support large abundances of herbivores and therefore much more complex food chains and food webs in terrestrial ecosystems. So one of the keys to understanding how these things work is how is it that they provide this kind of sustainable production that supports this large diversity and abundance of herbivorous mammals. And as I'm going to dig in pretty quickly here, when you start doing some of the accounting for how much nitrogen in particular is being input into systems or being lost from systems, there are some puzzling holes in the accounting. So if we sort of enumerate the typical nitrogen losses from tropical grasslands and savannas, so some recent papers published in just the last decade suggests that grazing animals actually serve, even though they recycle nitrogen, they often serve as a net export of nitrogen from the system because they move. And they often concentrate dung and urine in very specific places. So they literally spatially aggregate nitrogen. So from the majority of the landscape, the nitrogen is being contained in plants as they grow, and then it's just being moved. In places where there are cultures of livestock husbandry, the same sort of thing happens there because the animals are concentrated in what they call bomas or corrals at night to keep them safe. So literally half of their nutrient cycling occurs in a very small area. And a recent paper by Judith Sitters showed that that actually results in long-term reduction of nitrogen in organic matter as well as in plant tissues. But if we actually start putting some of the numbers on these things, so somewhere between 5 and 22 kilograms nitrogen per hectare per year are lost due to the movement by herbivores. And another, depending on the fire frequency, another 0 to 15 are lost from emissions due to burning. And then there's a few other processes like denitrification in the soil and so on. So when you add it all up, it's somewhere between 7 and 39 kilograms of nitrogen are lost every year. And keep in mind, there's lots of other factors affecting where you are in these ranges. And so the specific number is really not what's important. What's important is sort of understanding this range. So where would nitrogen come from? So one source is nitrogen deposition. In many tropical places, nitrogen deposition is actually really low. Because there's no nearby sources and nitrogen deposition is a relatively local process. Then, especially in tropical savannas, when there are trees, they tend to be dominated by leguminous species, such as what used to be known as acacia, which is now known as Vachellia. You know, the prototypical African landscape with the flat top tree. Okay, so that would be Vachellia tortilis, one of the more widespread species. So these are leguminous, and presumably they fix nitrogen. But nobody actually really knows how much nitrogen they fix. And so largely by subtraction from local nitrogen budgets underneath tree canopies, and a couple of studies in Australia where they did a nitrogen dilution experiment where you basically add labeled nitrogen into the soil and look to see what proportion of that shows up as opposed to other sources of nitrogen in the leaves of small saplings. There's a suggestion of somewhere between 0 and 4 kilograms of nitrogen are added by trees. But I would say that the large trees is a missing hole in this because people have recorded nitrogen fixation by large trees as deep as 3 meters underground. And so the problem is nobody's going to go dig up an entire tree just to make one spot measurement of nitrogen fixation. So people have tried to do it using various isotope methods, but those are sort of fraught with difficulty and hard to know if they're really working. So it remains kind of a big unknown. Then we have nitrogen mineralization, which is often stimulated by grazing animals because when they eat the plants, the nitrogen is put back in the form of urine and dung is rather rapidly decomposed by invertebrates and bacteria, and so often releases a lot of mineral forms of nitrogen that can be taken up by the plants. So there is some nutrient cycling effect. However, if I add up all those sources, then my range gets to be somewhere between 2 and 12. So what this suggests is that in many places, in many grassland savanna ecosystems, they may be out of nitrogen balance relative to what we know about it. So the challenge, and this is sort of a challenge I took up on myself the first time I ever visited Serengeti National Park, back in 2004 was, this is the hole that we have in trying to understand these savannas, is where is the nitrogen coming from, given that we know a lot of it is lost or moved. And so I want to first just kind of introduce the concept that guides me in thinking about how these things work. And I just call it the leaky bucket model. And so it is, it's probably the kind of accounting model that almost all ecosystem scientists use, but I find that it's useful because thinking about different pools of nitrogen, carbon, instead of being like some static thing, it's all very variable. And so the idea is that you have inputs, and then that there are sources of losses, and the magnitude of the losses can be represented by the size of the holes in the bucket. So if the holes are bigger, the same amount of input will lead to a lower standing pool of material. If the holes are smaller, then there'll be a larger pool of standing material. And the other thing is that I want to point out, and this is not necessarily a very common way of looking at this, is that the leaky bucket model actually implies a thermodynamic gradient. And that is that oftentimes the forms of materials that we track are often in the form of what I consider to be thermodynamically favorable molecules, meaning that they're relatively easy to transform into something useful. So these could be ammonium, sugar, or phosphate. So usually once these forms are used by various forms of life or undergo some kind of biogeochemical transformation in the soil, they are then converted into thermodynamically unfavorable forms, such as carbon dioxide or dinitrogen. So when we say these are unfavorable forms, what it means is that we have to add a lot of energy to the system to get those to be transformed into something more usable. Whereas the previous forms, we might have to add a little energy to get the reaction going, but it requires much less input. So photosynthesis requires solar energy because you've got to have all this extra energy to convert a thermodynamically unfavorable form of carbon as CO2 into something more thermodynamically favorable as the form of sugar. So the point that I'm trying to make here is not to get into the intricacies of the biochemistry, but rather get you to think about the fact that when we're talking about inputs and losses, we often just think about this as a one-way trip. But if systems are supposed to be sustainable, then we have to then have what would be the equivalent of pumps. And pumps, by definition, are a mechanism by which energy is used to convert materials or something and increase its potential energy. So pumping water up into a reservoir that lies above where it'll be used means that that water has high potential energy and can easily flow down through a plumbing system to be used. Okay, from a chemical point of view, the pump means that we're converting thermodynamically unfavorable forms of the chemicals into favorable ones. And so understanding what these pumps actually, how they might work, or thinking about them as pumps is kind of useful because the enzymes or the activities that are involved are special. So we'll talk about what some of those are. So, so the two transitions that I'm going to talk about today and talk about pumps have to do with dinitrogen getting converted into ammonium, which plants can use, or that ammonium then gets oxidized into nitrate, so it can be used, or photosynthesis, which converts CO2 to sugar. So what actually drives these conversions, and how do different factors on the landscape contribute to these kind of pumps being activated, or the strength of them, or the magnitude of pumping that goes on? So the work I'm going to talk about was done in Serengeti National Park. Now, many people think of Serengeti as these vast treeless plains with massive numbers of animals standing on them, and lions feeding on a carcass over here, and zebras running around over there, and wildebeest migrating in some big long line over yet some other part of it. Well, all that actually happens on a relatively small part of the landscape right around in here. The rest of Serengeti is immensely variable in terms of climate. The rainfall there ranges from 450 millimeters a year to over 1,400. Soil are also immensely variable, from soils in which phosphorus actually occurs in a higher concentration than nitrogen, to areas where phosphorus is measured in parts per million. So the park itself then provides this massive laboratory by which we can study how these different dynamics that I'm talking about, how these pumps might be really important in certain places and not in others, where losses are really high relative to inputs, because in some ways it encapsulates a lot of the continental level variation that we see across Africa, from extremely poor sandy soils to rich volcanic soils and everything in between. Okay, so starting in 2004, I wrote a grant where we were going to focus on measuring nitrogen fixation in a dominant legume that occurs in the system as kind of a starting point. First of all, to work out the methods for measuring nitrogen fixation, and then finding something like some low-hanging fruit, as it were, and try to understand variation in nitrogen fixation across this interesting landscape. So after I got that first grant in 2006, and we made these measurements, we were indeed able to measure that this legume was able to fix nitrogen, and that the amount of fixation was varying across the landscape. So then we decided to sort of beef up what we were doing and look at broader patterns of variation in nitrogen fixation in relation to nitrogen and phosphorus gradients. So we were going to sample in a lot more places and do more duplicate samples. But this is where Nancy Johnson came in and said, you know, I've been reading these papers that seem to say that grass fixes in. I think we should at least measure it and see if we can pick it up. And I'm like, well, okay. I mean, we can only afford to ship in this many bottles of 15N2, but I'll try it. So then I had to decide, well, gee, which one of these species should I pick? So through a complicated set of things, I ended up picking one species called Themata triandra, which is a pan-tropical grass. It occurs in Australia, Indonesia, India, all across Africa, and then it's been naturalized in Brazil. It also forms monospecific stands and seems to like waterlogged soils. So at that time, I'm thinking waterlogging had something to do with whether nitrogen fixation could have happened. So I just kind of picked it as this seems to be the most likely candidate. If we are going to find it, we'll find it with this species. Okay, so indeed, when we did measure it with Themata triandra, we did find that it was fixing nitrogen, and that rate at which it fixed nitrogen was variable across space, but we weren't able to look at it in very many places the first time around. So these data represent another iteration of effort in which we tested whether nitrogen fixation was occurring in 6 different grass species that are all relatively common, at least across 50% of the park. So they're very widespread, common species that tend to show up in all your samples. They have very different, how should I say, life histories. They're all perennials, but some of them are like heavy grazing tolerant species like Digitaria macroblephera, the one here all the way on the left. Panicum maximum is a grass species that grows up under, is often found underneath savanna trees. Pennisetum mesianum forms monocultures on volcanic soils, oftentimes. So the point is these represented a kind of a wide variety of different kinds of grass life histories. And then as a reference, I also included our friendly legume, Indigofera fulkensii, which occurs at about 70% of our sample points across the park. So it can deal with heavy rainfall or relatively little rainfall. It's tolerant of grazing. So you find it a lot of different places. So what these data are here is a measure of the copy number of the nitrogenase gene in the plant, in the root tissues. So the idea here was, well, are we even picking up nitrogen fixing bacteria in these plants? And so the answer is yes, we're picking that up, but some of the species have orders of magnitude more greater abundances of this gene copy than others. So this would suggest that some of these might be fixers and some might not. And keep in mind that that number is never going to be zero, because the old adage, bacteria are everywhere, but the environment selects. So they might be found in plant tissues, but they're just not active or supported by the plant. Okay. So then we actually did our 15 into uptake. So basically the way that works is that you take a root and you rinse it off, but we've also done it where we did surface sterilization so that we would get rid of any signal of the soil bacteria that might be contributing to the measurement. But most of the time we just rinsed off the roots. And then you basically put them in a big test tube, you seal it, and then you add your N15 gas to create a certain atmospheric percentage, and then you incubate them in the soil. And most of the times the soil temperatures were somewhere between 22 and 25 degrees C. And then after a certain time, you also do this with a control plant that you don't enrich the atmosphere for. And then by comparing the N15 in your, where you've enriched the sample with your control assay, you get an estimate about how much nitrogen was from the atmosphere was accrued in the tissue of the enriched samples. So based on this method, we found typical distributions of points. There's often quite a few zeros, but then also some really high values. So none of these things are like super normally distributed. So normally when we're actually analyzing the data, we end up doing some transformations on this to make them normal. But the bottom line here is that 3 of these grasses, so that would be, so Digitaria macroblefera, which is the most common grazing tolerant grass in Serengeti. And then the legume Indigofera fulkensii, and Pennisetum mesianum, and then the one that I luckily tried the first time around, Themata triandra. All of these show significant nitrogen fixation rates across all the sites where we measured it. They're significantly different from 0. There are 3 other grass species, Ergrostis tenuifolia, which is a very small, shallow-rooted grass that's mostly interested in making seeds. So it really acts more like an annual than a perennial. Panicum maximum, which is a very tall grass that likes to grow where there's higher water availability or other things that make it like to live under trees. And then Spirobolus fimbriatus, which is another extremely common grass, reasonably tall. So there's maybe some of the samples showing some evidence for it, but this is not significantly different from 0. And these 2 were clearly not different from 0. So the conclusion that we drew from this is that some of the species of grasses show the capability of fixing nitrogen. The important thing to understand is that a lot of this variation in rates is also driven by where we measured it. So we'll talk a little bit about that in a minute. The other thing to notice, and this we found out much later when we actually started making the measurements, is that the critters involved are putative nitrogen fixing bacteria that we found in the roots that were most common, were somewhat different species than what most people have reported when they reported in the bacteria composition of grass roots. And it does share in common some common species that are found in soil, so free living soil bacteria that fix nitrogen. But the interesting thing is that even the legume, the most common bacteria, putative nitrogen fixer in legumes was Enterobacter, and it was not Rhizobia. Furthermore, Indigofera, even though it's a legume, does not form nodules. So all of this stuff, given that we surface sterilize roots, is being done by endophytic bacteria that are just living in the root. The other thing to notice about these species is that they all have adaptations for isolating the nitrogenase within the cell. So they have various little internal structures. So they don't require nodules because they have their own ability to avoid oxygen, which competes heavily with dinitrogen for the nitrogenase substrate. So this is kind of like a whole new world. Because first of all, most people never think about grasses as fixing N. Two, even legumes are supposed to be doing the old-fashioned thing and making nodules and using Rhizobia. And three, some species are doing it and some species are not, and we don't really know why, just because this is like the first discovery. So many questions about this remain. And if you have some questions, happy to ask after the talk. Okay, so then the first thing that comes up is, okay, well, you did a bunch of short-term assays and you found that sometimes there was nitrogen being fixed, and okay, that's fine, but is this really persistent? Well, short of doing 10 consecutive assays over a month on essentially the same plants, the better way to get at that is to see if there are isotopic signatures in the plant tissue that would be consistent with persistent nitrogen fixation. So in this way, so what we did was compare the rate of nitrogen fixation in the sampled root with the difference between the N15 and the, this says the leaf, but it's supposed to be the root. The root N15 and the soil N15. Okay, so and if you put all of the data together, they all give you a significant regression with a negative slope. The point is, the more negative this is, the greater the departure between the N15 in the plant relative to that in the soil. So presumably, if you're not fixing nitrogen, the N15 would be the same as being in the soil, so you would be around 0. So clearly, some of the plants we measured are around 0, which would be consistent with them not fixing nitrogen. But the bigger this deviation, the fact that these are correlated suggests that when we had a short-term measurement of nitrogen fixation, it was often from a plant that showed a strong deviation in its nitrogen 15 signature in the tissue. So, but interestingly, there's a lot of variation among the species in how strong this correlation is. So in the legume, we had a very nice correlation there. There's still a correlation, but it's not nearly as strong for Digitaria. And then Pennisetum actually shows also a very strong correlation, a bit weaker for Themida. But again, as you look at the data, you see that it's the only time you're really getting high rates of nitrogen fixation. They are from plants that have a strong deviation from the soil N15. The other point about this is that where Themida is abundant and occurs, the N15 in the soil is often lower than it is other places. So it's just a statistical artifact. So from this, we can conclude that the short-term assays we measure provide suggest that the plants that we're measuring them in are fixing in consistently, repeatedly, persistently over time during growing season. All right. So this gives us two opportunities to estimate, like, so how much nitrogen are they actually fixing? Is nitrogen fixation really even possibly, given these rates, is it really possible that it's making up that difference in nitrogen that we pointed to initially? And so we actually calculated this in two independent ways. So one way was we took our local rates and then we just multiplied. So we multiply them by, so we had a fixation rate per minute. So then we multiplied by minutes per hour, hours per day, days per season. Okay. Then the other way we did it was to use the N15 data and use it as a way of estimating what percentage of the N in the plant came from, so you can estimate what percentage of the N in the plant came from atmospheric sources based on that deviation between the soil N15. And then we also, as a check, used the N15 in the tissues for the same species for those that we didn't see any, that where we detected no nitrogen fixation in the assay, even though there's a little risk with that because they could have been fixing in at some other point in time. They just weren't doing it when we measured it. Okay, without getting into the gritty details, both methods gave relatively similar estimates for annual nitrogen fixation. So what were those numbers? So we measured this at 5 different sites across the Serengeti. Would have loved to measure it at more, but we only had so many assays we could do, and we're having to split them amongst the 6 different species. So we're like doing bioprospecting at the same time we're trying to measure an experiment, and we just sort of ran out of samples. But a little bit of information about the sites might be helpful. So this SOT site is the driest site. Gets about 500 millimeters of rain a year. This is the Serengeti Wildlife Research Center, which gets about 600 millimeters a year, and the soil is kind of sandy, mostly nitrogen limited, has higher than expected phosphorus at it. This one has yet more, slightly more rainfall, but it's functionally very similar to the SWRC. Then this site here is extremely phosphorus poor. Has high rainfall, like the soil nitrogen phosphorus ratio is about 40. And in many places, the phosphorus is measured in somewhere where 2 digits parts per million. So it's very low in phosphorus. Then this site, Musabi, is also a heavy rainfall site. So both of these sites are heavy rainfall. But this one has high levels of phosphorus, this one does not. So one has to be careful trying to make any kind of inferences about functions by just comparing 5 sites. The point is that we did see variation among both the species. Where they showed peak nitrogen fixation seemed to be at different sites, suggesting there's sort of almost like a functional ordination of nitrogen fixing plants. So the one that's providing peak nitrogen fixing at the driest site was Pennisetum mesianum. So that was that grass. Then Digitaria was peaked at a different place, and then Thamida triandra peaked at this site here. But the more important thing about this graph is just simply that these estimates of annual N fixed measured as kilograms nitrogen per hectare per year are all ending up somewhere in this middle of a box that's representing this range of potential losses due to grazing and fire, which we discussed earlier. So the fact that each one of these little histograms is somewhere in the middle of this range is consistent with the idea that nitrogen fixation by grasses is contributing towards the balance of nitrogen in these systems. And that effectively the nitrogen fixing bacteria in these grasses are essentially pumping nitrogen into the system and are contributing to its sustainability. Okay, and so this is just a graph to show you that the nitrogen fixation also seems to correlate with leaf tissue element concentrations. Again, circumstantial evidence consistent with the idea that they're a pump. So on the x-axis, we have the log of nitrogen fixation on a per gram of root basis. And then this is leaf N in the same plant. And then here we have the phosphate activity in the soil as a function of leaf P. And so the phosphatase activity, so the important thing to know is that even when there was a low amount of this, there was a big range in leaf nutrient concentration. When it was high, then that range was much lower. So the same thing was true for nitrogen. So it suggests that there's some influence on these various rates as to plant nutrient status. So what does that mean? Well, if we're thinking about how do I support a large abundance and biomass of herbivores in the system, I need nutritious plants. And an interesting comparison is if you take the global database analysis that Peter Reich and Jacqueline Oleksin looked at back, it was a paper from 2013 in I think it was PNAS, if I remember right. The global grass leaf N for C4 grasses, since all of these grasses are C4s, is at the bottom end of this range. And the same thing is true for the phosphorus as well. And the interesting thing is people, like if you've read all the papers from North America that talk about C4 grasses and what their traits are like, what their characteristics are like, they're often depicted as these large, low nutrient, unpalatable grasses. But in Serengeti, the leaf nitrogen for a significant portion of these grasses sampled is above 2. That's the same level of nitrogen that you get in fertilized grassland on your lawn. Okay, so that's just providing some perspective on what the magnitude of these different things is. So even though these are purportedly low nutrient grasses, they actually have nutrient contents that are much higher than you would expect. Okay. So now I'm just going to shift gears a little bit, and then at the end, I'll come back and try to put these two stories together. So another big feature about Serengeti that's unique is that the place has about 2 million wildebeest, and these wildebeest do a large scale, roughly 400 kilometer in total migration on an annual basis. That's been, I think, every single documentary that has been done about the system Serengeti or the Mara National Reserve in Kenya always talks about the wildebeest. Now people want them to see being eaten by crocodiles rather than migrating across the plains, because that's much more interesting. But the important thing is that they move, which means that when they come through an area, they graze it very intensely and then they move on. Because of this, there's an entire movement in the regenerative agriculture sector to recreate this kind of movement using what's called rotational, short duration, high density, adaptive multi-paddock. It has about as many labels as you can imagine. The idea is that you're trying to simulate a migratory system by moving our animals around in space. So the argument is that this is benefiting or has the same beneficial effects as migratory grazing by wildebeest. And it's kind of like, okay, but exactly what is that benefit? So the dynamics that we're talking about, I just want to make it clear. is the difference between so-called continuous grazing. So the idea here is that the plant, let's just say grass, the grass is being consumed repeatedly over short periods of time so that it's maintained in a relatively short state. There's tissue being lost all along the process. And then there's nutrient cycling going on almost continuously, as opposed to a what I call an episodic grazing situation where plants have some period of time which they grow up. Then there's a consumption event that reduces the biomass down to this level. And then eventually and I'm showing two species here because in Serengeti, it was famous for having a succession of species. So first one comes and eats all the coarse plant material, then another species comes behind it. It's really not quite that tidy, but the point is there's a phalanx of multiple herbivore species that come through. Then there's a regrowth, okay? And then juxtaposed to that is the idea of, well, we have ungrazed conditions. So what happens if you just take the herbivores out of the system? So then during a growing season, you would grow from a short stature to a tall stature, and then it just kind of sits there in a rough, steady state where growth is still growing, but the plant tissue is sort of dying at the same rate, or the amount of carbon coming in is roughly the same as the carbon coming out through respiration or tissue turnover. Okay, so that's kind of a description of what I mean when I'm talking about migratory or episodic grazing versus a continuous or conventional grazing. And like I said, most ecological models treats herbivory like this. It's treated as a continuous process. The term is actually like a rate equation. Change in biomass over change in time. Okay, so why does this matter? Why would it matter? So it occurred to me that if we want to understand the effects of this episodic grazing, we need some kind of a logical mathematical framework to look at it in. So it turns out that the easiest way to set this up is to assume that I'm looking at like a relative growth rate of the whole community. So this is not a relative growth rate of an individual species. It would be like looking at the growth rate of all the species at the same time. So I call it a community relative growth rate. But essentially it's a new mass of plants per mass of plant that was there before per unit time. All right, so the assumption is that plant growth is ultimately, in the absence of grazing, is ultimately limited by some resource. We don't know what it is. Doesn't really matter too much what it is. But the idea is that there's a density dependence, meaning the greater the biomass of the plant, the lower its relative growth rate, because of essentially there's too many resources tied up in the actual biomass and that aren't available to be converted into growth. So this is just being modeled as a simple logistic equation. But then we assume that some biomass, which we'll call the initial biomass here, then grazing occurs, and there's a reduction in the biomass down to this point, which then shifts the community from growing at this rate to now it's growing at this rate. So when you model that, if you plot biomass over time from the same starting point, then it would have grown like this into the future if it was not being grazed. After the grazing event, it's growing pretty much like this. So it turns out that you can then calculate what's the actual production that you get over the whole time, which includes the growth of the grass before the grazing event, including the loss of the grazing event, plus the growth of the grass after the grazing event. So to make a long story short, it makes a prediction that under a wide variety of conditions and without any nutrient cycling whatsoever, you can get a stimulation of production at intermediate intensities of grazing. So this herbivory intensity here is like what percentage of the standing biomass was removed during the event. So the point is that these intermediate grazing intensities, you can get a stimulation of grazing of production by as much as 60% without any nutrient cycle. So this is kind of revelatory in the sense that the only reason we know that this can happen is just because we broke down the logic of the process of a grazing event relative to what the plant would be doing if it had a certain biomass or if it has different biomasses. So then we can also define sets of conditions under which we can see this. So does this occur only under some weird combination of parameters, or does it occur under a wide variety of conditions? So it turns out that the initial biomass has a lot to do with whether or not you get the stimulation of production. And we measure this as kind of biomass that's present at the time of the grazing event relative to what the biomass would be in the ungrazed condition. And then the other thing that affects it, of course, is how intense the herbivory is. The point is that there's this zone of grazing intensities and initial biomasses at which you would get increased production. So does this model hold any water? I mean, it's one thing to write down a bunch of equations. Question is, does real life do anything like what the equations would suggest can happen? So I went back to Sam McNaughton's original data that was actually collected in 1973, but he didn't publish it until 1985 in an ecological monograph. So I had access to his raw data, so I went back to his raw data, and then when we looked to see the comparison of production at a whole bunch of sites across Serengeti. So some of these are high rainfall, some are low rainfall. The point was that they all varied, and they also varied in their grazing intensity. So some were heavily grazed, some were lightly grazed. And so what we found was that there was evidence that the ratio of the biomass at the time of grazing relative to the biomass in the ungrazed component. So that means the farther up this axis we go, it was the higher the initial biomass at the time of grazing. So we can see that this production event went up as you had higher and higher initial biomass. And this makes sense because you're giving, think about it as if you're freeing up resources for growth, you're going to free up a lot more resource for growth if you didn't have many to start with, which occurs at the high biomass concentration. Okay, then we also used the model that I did to predict the change in production relative to ungrazed. So we can measure production in the ungrazed, and then we can measure production in the grazed system. And so anything positive here means that under grazing, you're more productive than you would be if you didn't have any grazing at all. And so the R squared isn't super high, but there is a significant correlation and definitely a trend in the data that more or less matches up with what was observed. And then we can also predict whether or not we got this curve pattern that I showed you previously. So let's go back. So remember this one. So we predict this curved pattern that as you increase grazing intensity, you should get an increase in the stimulation of production followed by a big drop off. And so when we looked at it, then we see that same pattern happening here. And the point of all of this mess is just to show that when we look at the data, if we account for both the initial biomass and the grazing intensity, and we allow for the hypothesis that there's a curve, so we modeled that as quadratic, all of those factors were significant at the same time. Okay. So this suggests that the dynamics that Sam McNaughton wrote about and was so controversial, that grazing and the way people interpret it, grazing is good for plants, is supported even without any additional benefits of nutrient cycle, short-term nutrient cycling. And that it can be explained by this interaction of episodic grazing with density-dependent plant production. Okay, so what happens when we put the stories together? Because what's really being suggested by the stimulation of production is, in fact, grazed systems are capturing more carbon. They're fixing more carbon than an ungrazed system under certain combinations of conditions. So one way of looking at this is do an experiment in which we measure nitrogen fixation rates inside exclosures or outside exclosures. So the standard dogma on this is that if we have grazing, that reduces the amount of photosynthetic tissue, which presumably reduces the amount of carbon that the plant has to allocate to beneficial microbes that might be fixing nitrogen. And so grazing should cause nitrogen fixation rates to go down, because the idea is that they're become carbon limited under grazing. The alternative hypothesis is that grazing stimulates the assimilation of carbon in the system. Then it might also stimulate nitrogen fixation because there's more carbon available, not less. So we looked at this then in two experiments. So one was just a regular grazing exclosure experiment, and this was from the first round of nitrogen fixation measurements, where I was only looking at Themata triandra. And what we found was that, this is now measuring nitrogen fixation rates per unit area. So it's accounting for the biomass of the roots of the plant species in the environment. And so what we found was that for Themata triandra, there was not a significant difference in grazing in the rate of nitrogen fixation per unit area at one site, but there was at Kleinskamp, and this is the site where Themata was fixing the most, by far the most nitrogen. By comparison, if we look at the legume, we saw really no effect of grazing on nitrogen fixation by the legumes, which are eaten, by the way. So even though they're not grass, they're still consumed by grazers. Okay. Then we had another experiment where we looked at nitrogen fixation under different fertilization regimes, and there's really no huge surprise here. Adding fertilizer basically took away nitrogen fixation. That's not a big surprise. What was a surprise is that for the grazing tolerant grass, grazing stimulated nitrogen fixation. For Themata, there was no difference. So obviously there's some variation across sites that seems to contribute to exactly what effect grazing has on nitrogen fixation. But what it does suggest is that there's a coupling between the carbon dynamics as driven by things, episodic grazing, climate, soils, and the nitrogen dynamics, and particularly the nitrogen pump of nitrogen fixation, that those two are interdependent in some way. But we don't yet know what that is. So just to kind of summarize what we have is there's evidence of substantial nitrogen carbon losses due to fire and grazing from these savanna tropical grasslands and savannas. It's not very clear, although there's still a huge hole in the data, a bit that trees and herbaceous legumes can make up the difference. But for it to be sustainable, we have to have some kind of pumps in the system. So they could be trees, they could be something else. But what we discovered in Serengeti was that grasses are essentially acting as those pumps in a mutualistic association with endophytic nitrogen fixing bacteria in their roots. There's a lot of spatial variation because the local conditions of soil, climate have a big influence over the strength of nitrogen fixation. And then those outcomes are coupled with the fact that episodic herbivory in this migratory system seems to stimulate production and therefore providing more carbon to the system. And that there's a complex coupling of these two pumps, nitrogen and carbon, that we need to figure out more. So that's what I've got for today. So thanks for listening. So happy to take any questions.
Yeah, great. Well, that was a fantastic seminar, super cool results. We'll take maybe 1 or 2 questions from the audience here, and then we can go to Zoom.
Yeah.
This happened with grasses that they died in Serengeti.
Yeah. So that's always the first argument. Well, Serengeti's weird. Yeah. Oh yeah. Okay. Sorry. So the question was, does this happen anywhere else other than the Serengeti? So the answer is yes. The stimulation of productivity is highly climate dependent. So in places that are cold and dry, the level of production that can occur after you get an episodic grazing isn't high enough for you to overcome the losses due to herbivory. But in more mesic systems, like for example, there's like an increasing abundance of data from places in the Midwest and the East Coast of North America, for example, that these kinds of mobile grazing effects can have massive increases in the carbon coming into the system, sufficient that it can cause soil carbon to increase. So it increases the pool of soil carbon that's fed by roots and decomposing plant tissues. I didn't really get into the soil carbon story today, but it is linked to this. Yeah, that we don't know. There is evidence that switchgrass, which is a common biofuel, so it's been pretty heavily studied, that switchgrass, but it's also a very common tall grass prairie species, that it does fix nitrogen when it's growing in nutrient poor soil. And so but nobody's measured it yet. Yeah. Right. Yeah. Okay. The question is, if people actually did go out and measure it, do you think they would find it? And my response is, I'm pretty sure that if we go to other tropical systems, we will find it. And the evidence for that is that if you look at the N15 signatures of the root and leaf tissues of especially tall grass species that grow in nutrient poor soils all across Africa, their N15 signatures suggest that they're persistent nitrogen fixers. And that would be similar to the KCW site where we found that Themata was fixing so much nitrogen. So in the tropics, I say yes. In temperate zones, there's a complication of temperature. Nitrogen fixation is very energy dependent. And if soils are too cold, the rate of nitrogen fixation is likely to be reduced sufficiently that it's not that important or not able to overcome things like losses of nitrogen to fire. So, but again, we don't know, because microbes are immensely adaptive. And so there may be suites of microbe species that can fix N, but they do it at a cooler temperature than what people are normally studying, which is microbes in crops. So that's another caveat. And most of what we know about nitrogen fixation is in crop species growing in crop. So it's annuals growing in tilled soils. So, yeah, question. Okay. Okay, yeah, that's good. Okay, so the question is, how do you determine if those bacterial groups are actually responsible for fixation? That's a very interesting but tough to answer question. Typically, bacteria that fix N don't do well when you try to culture them separately, so it's really hard to see. Also, even if you did that and you found they were fixing N in the petri dish, doesn't necessarily mean that they were doing it in the plant. So The only way I know to really try to get at that is to just do a massive amount of sampling and then try to do it through some kind of analysis, statistical analysis, or even like a machine learning analysis where you try to figure out, where you try to portion where you had lots of fixation, what was the probability that you had a certain bacterial species being very abundant. That in my view would be the only way to do it right now. But eventually we may find ways of where you can like doing some like gene editing and things like that to see whether or not you could do it. But even then, it's still going to be done in a very sort of sterile environment that's not really linked to the field. So, but a very good question. Thank you. Yeah, so she's asking if you did the like detailed gene sequencing, whether you can detect by looking at the sequence of the genes, whether or not they were more or less likely to be fixing. And I don't know the answer to that.
Great. We are out a little over time, so if anyone needs to leave, we totally understand. I'm going to ask one question from Zoom, and that's why do you think that some grass species appear to fix and while others don't?
Yep. Okay. So the question was, yeah, right. So why do some grasses fix? I think there's different reasons for different grasses. So let me just start with the one grazing tolerant grass. So so its whole strategy in life is to be grazed and then regrow really fast. In order to do that, it has to have plenty of resources with which to grow. So and it's also always living in an environment that's not particularly light limited because it's just been grazed. So it may be that nitrogen fixation is its answer to, I need resources and I need them now because I have to grow fast. Because if I don't, then all those other plant species that also got eaten have the advantage over me, if I can anthropomorphize the explanation. So Femida, on the other hand, lives in environments with higher rainfall and lower phosphorus. And it may be that it fixes N, or one way of arguing it is that evolution of grasses that fixed N end up leaving more N behind. And so that it essentially prevents the degeneration of N from their local root system or whatever. The other argument is the hypothesis that nitrogen fixation is also linked to the phosphorus cycle because the nitrogen that's used from fixation can then be used to synthesize enzymes called phosphatases that are then exuded into the soil and then can release phosphate off of minerals or off of organic matter. And so it's that plant's answer to phosphorus deficiency. So these are all great questions, and we don't know the answer to any of those things. And the whole idea that grasses might fix nitrogen for different reasons to adapt to different conditions is a totally new concept. Yeah. Yeah, thanks for listening.